Good evening, everyone. Let's start the class. SEC paper. This class allotted for SEC paper Saturday. Semester 1 SEC paper has food fermentation technique and packaging. Food fermentation technique and packaging. Let's look syllabus first. What are the units have in the syllabus? Syllabus total 8 units. Consist of total 8 units. Now segregate those units. Unit 1. Unit 1 contains fermented foods. Unit 2 to 5. From 2 to 5 units, all has fermented food product production. Fermented food product production. We have milk as a food product fermentation, cereal based, grain based, etc. etc. From unit 2 and 2 to 5. Unit 6 contain probiotics. Unit 7 has controlling microbiological quality of the food products. Microbiological food products. And unit 8 is about packaging, food packaging. The overall name of the SEC paper is food fermentation technique and packaging. Packaging is in the unit 8, food packaging technique. Actually, it is a part of food microbiology, not overall food microbiology. It's taken from the food microbiology and created this syllabus. Okay. Question paper from previous year. This is the syllabus you have to do, total 8 units. This SEC paper is only theoretical, no practical based. It is completely theoretical and total marks of theory 100 marks. Total marks 100. But previous paper, core paper has practical 25 and 75 theory. Here total theory marks and it is 100. Marking segregation same as it is, like 2 marks question, 3 marks question and 5 marks question, but number of questions is increased here. 2 marks question have to be answered 10, but it contain or total question present 15. Out of 15 you have to be answered 10. So here it is 20 marks total. 3 marks question also 10 question have to be answered. Here also remain or internal or 15 question total. Out of 15, 10 question, that means total 30. And 5 marks question total have to be answered 10. and internal odd and total quotient 12, 50, so total 100. That is the marks distribution. Question 2 marks, 3 marks, and 5 marks. Highest awarded marks 5. Sometimes total 12 question and have to be answered 10. Option come 5 marks. 3 hours. 2 hours. So highest awarded marks 5, sometimes split also there, 2 plus 3, 3 plus 2, etc. etc. Above 5 in Calcutta University, very less question. In case of SEC paper also. So that's all about syllabus and question paper. Now came to the book suggestion. As we earlier tell about that, it is a part of food microbiology, not the complete food microbiology. So there is no specific book for this paper, no specific book. But I will suggest some book, but have to stick my notes and class notes for this paper preparation. No specific book, collectively no book available for all the syllabus that maintain. But two books are better for this paper study. Number one, Food Microbiology. You can purchase this book. Food Microbiology by Frazier. It is Tata McGraw production publication. And number two, Fundamental of Food Microbiology. Vivek Ray, CRC Press, Indian author. And other is Frazier. I repeated these two books not completed the all topics. Clear? So no books for this specific books for this paper. So must stick with my class notes and the notes that is given after unit completed. That means after unit notes. That will be beneficial to prepare this paper. And you can see the book that is suggested. All are food microbiology book, and the topic is here food fermentation technique. That means it's some parts are taken from this syllabus, food microbiology syllabus, and make this unit in this paper, SEC paper. Clear? Book suggestions, syllabus, question paper analysis, etc. To start with this topic, first topic, unit 1. Fermented foods. Fermented foods. And today we are going to discuss the basics about fermentation. Today we are going to discuss the about fermentation. Tell me the word fermentation means. What do you know about the fermentation? You read about this fermentation in previous class. So answer fermentation means. The dough gets fermented by adding milk. That is the process. The actual meaning of fermentation. Online anyone? You can answer in any language. Bengali you can answer. But don't be timid answer. Everybody try to answer the question. Otherwise concept will be not clear. What is the meaning of fermentation? It's very basic fermentation. Can you write this statement? Fermentation is the process of energy production step in some microbes. Can we write? Is it correct statement? Is it correct statement? It is a part of energy processing, energy production step in every cell. Not every cell, in case of microbes. Can you write? So energy production step. Fermentation is production, create energy. So you all know that all cells, all living cells need energy. All living cells need energy. And where does the energy get from? From the food products. And the energy come from the food products. Because food products contain energy, internal energy. You have to be metabolized and capture those energy content in the food products. And the process is called metabolism or respiration. So how living cells get energy from the food products? By the means of respiration. By the means of respiration. And we know respiration can be of two types, aerobic and anaerobic respiration. Respiration can be of two types, aerobic respiration as well as anaerobic respiration. What is the basic difference between aerobic and anaerobic respiration? Aerobic requires oxygen. Oxygen. Anaerobic does not. Does not. Okay, good. Aerobic respiration needs oxygen to break down the food products to capture those energy in the food. Need oxygen. But anaerobic food respiration doesn't need oxygen. So fermentation is categorized into aerobic or anaerobic food production mechanism. It is categorized in anaerobic food production mechanism. So fermentation categorized inside anaerobic respiration. Fermentation is a type of anaerobic respiration. Is it clear to all? So what is the basics of fermentation? What is the meaning of fermentation? It is the energy gathering state. Every cells need energy, and for this cells capitalize food products and enter into the respiration. Respiration can be of two types based on the situation. If oxygen is absent, then anaerobic respiration, and the presence of oxygen, it will be aerobic respiration. Aerobic respiration done by most of the animals, plants are done aerobic respiration. Most of the plants and animals, what they do? They do aerobic respiration. Anaerobic respiration done by microbes. Not all, but microbes can done anaerobic respiration, including fermentation. Microbes means bacteria, some bacteria, as well as yeast from the fungi section. Yeast can do fermentation also. Did you know sometime animal cell in some situation they are doing anaerobic respiration, that means fermentation? Can you give some example? When athletes Olympics are there. Olympics are going on. So athletes are requiring energy for 100-meter race, for sprinting. So when the animals or some humans are sprint, that means quick burst of energy are required for sprinting or during heavy exercise. A quick burst of energy is required, but that time oxygen getting very low inside the tissue. So the situation is two. During the sprinting or heavy exercise, two situations are created in the body. Oxygen concentration that time down, and you need more energy to carry on the process. Yes or no? Yes. So that time muscle, skeletal muscle, do anaerobic respiration because oxygen getting low inside the tissue and start doing anaerobic respiration. So in certain situation, animals, that means humans, the highest order animal, can do fermentation, but most of the time they are doing aerobic, and microbes like bacteria, some bacteria and yeast always do yeast, sometimes aerobic, sometimes anaerobic based on the situation, but some bacteria always do anaerobics because they prefer to live in anaerobic condition. So start doing anaerobic respiration, and they are getting habituated with that. Clear? Online. Those are in the online. Clear?
Yes, sir.
Okay. So whatever the respiration, it will be aerobic or anaerobic. The common intermediate, common state of aerobic and anaerobic before enter into aerobic or anaerobic is the glycolysis. Did you hear the term about glycolysis? So aerobic, anaerobic, whatever it is, the glycolysis is the first step before it enter. It has two options. It can enter aerobics or anaerobic. So then we discussed about the glycolysis. The next topic come, that is glycolysis. Because in all scenario, aerobic respiration and anaerobic respiration both require the common state, that is glycolysis. Glycolysis means breakdown of glucose. Breakdown of glucose. Yes, we heard right. Glucose, that means carbohydrate, is the preferable source of any energy requiring metabolism. Cells try to produce glucose from any source, fat or protein, try to convert into the glucose or carbohydrate basics, and then it start to produce energy. So in case of bacteria also same, they prefer glucose, that means carbohydrate source, for the energy source, and it enter into the first step, that is glycolysis. And glycolysis is the chain reaction state catalyzed where glucose is being break down and a convert into pyruvate. The second point, in glycolysis, it is chain enzyme catalyzed reaction state. So in glycolysis, how many reaction steps are there? Total 10. And all reaction states are catalyzed by the enzyme. 10 enzyme catalyzed reaction state. Glycolysis occur in cytoplasm, cytosol, and for glycolysis do not need any oxygen. That's why glycolysis is the first step in any scenario of aerobic or anaerobic. Do not need any oxygen. So glycolysis happen independent of oxygen. Independent of oxygen. Glycolysis has two phases. A total 10 reactions catalyzed step are divided into 5. The first 5 steps are called preparatory phase. Preparatory phase. Preparatory phase is the first 5 reactions. And the next 5 reaction called payoff phase. Payoff phase. Last 5. So we divide the total 10 reaction catalyzed into half. First 5 and 5. First 5 is called preparatory phase. That means here enzymes catalyze the glucose molecule and prepare for energy production. The payoff means, what is the meaning of payoff? That means it given something. So payoff phase, the ATP will be produced. First 5 reaction is the preparation where glucose is modified and it is prepared to form ATP now in the last 5 reaction. That's why it's called payoff phase. 2 phase is there in glycolysis, the intermediate step 5 and 5, preparatory and payoff. Clear? Check the reaction steps in glycolysis. We started with glucose. Glucose first converted into glucose 6 phosphate. Glucose 6 phosphate. In glucose 6 phosphate, what is the change in the glucose structure? A phosphate group is added and the glucose is 6 carbon molecule. If you look the structure chemistry of the glucose, it has 6 carbon and the number 6 carbon, the addition of phosphate done. That's why it's called 6 phosphate. Glucose 6 phosphate, that means carbon number 6 contain now a single phosphate group. And this reaction state need a ATP molecule. A ATP molecule, because ATP contains 3 phosphate, adenosine triphosphate, tri means 3. ATP contains 3 phosphate. One of the phosphate is transferred into the glucose molecule in the 6 carbon and produce glucose 6 phosphate. And now ATP will become ADP. From tri, it become di, ADP, adenosine diphosphate. And this reaction state catalyzed by hexokinase enzyme. This reaction state catalyzed by hexokinase enzyme, and the short form of hexokinase HK. It will be better to remember in the short form HK, hexokinase. After that, glucose 6 phosphate convert into fructose 6 phosphate. Convert into fructose 6 phosphate. Actually, in this step, no changing of the phosphate group occurs. Just change the structural molecule changes. That means glucose is aldehyde containing sugar and now it convert into keto group containing sugar, fructose. That means here isomerization occurs from aldose to ketose. Aldose is glucose and ketose is fructose. So aldose to ketose just changes the molecular structure, overall formula, do not change the phosphate group. So it is a isomerization reaction and the name of the enzyme also related to the isomerization. That means the name of the enzyme glucose phosphate isomerase or it's called phosphoglucoisomerase. Phosphoglucoisomerase. And the short form is this PGI, phosphoglucoisomerase, that convert glucose 6-phosphate into fructose 6-phosphate. In next case, fructose 6-phosphate convert into fructose 1,6-bisphosphate. Glycolysis also there in the class 11 or 12 syllabus, but I think here details, that much details is not. Just overall reactions are there. But here we should retain about the enzymes as well as cofactor, etc., etc., and know the reaction, what reaction is going on. That means glucose to glucose 6-phosphate, that is transfer reaction, phosphate transfer reaction, glucose 6-phosphate to fructose isomerization reaction, etc. So that details in the BSc syllabus. Fructose 6-phosphate convert into fructose 1,6-bisphosphate. So one phosphate means another phosphate group is attached. Already there are 6 carbon position, there is a phosphate and another phosphate is came and joined in the carbon number 1 in the fructose and form 2 phosphate group now. That's why you written here bis 1,6-bisphosphate. Enzyme name phosphofructokinase. Phosphofructokinase. Short name is PFK. Short name is PFK. Here also same scenario, that is reaction number 1. That means here also another ATP molecules are required to transfer the phosphate in the carbon number 1 of fructose. So ATP to ADP, done. Reaction number 3. Reaction number 1, 2 and reaction number 3. Fructose 6-phosphate now break down. Fructose 6-phosphate break down into dihydroxyacetone phosphate. The short name of this DHAP, 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 dihydroxyacetone phosphate. And another molecule, glyceraldehyde 3-phosphate. Short name is GAP. So fructose 6-phosphate just break down evenly, split from the middle and form GAP and GAP, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. We started from the glucose. Glucose is 6-carbon structure, 6-carbon 6C, glucose 6-phosphate 6C, 6C, fructose 1,6-bisphosphate also 6 carbon, but when it split, it becomes 3 and 3, 3C, 3C. Next reaction, this DHAP molecule, dihydroxyacetone phosphate, convert into GAP, glycerol 3-phosphate. So no DAP molecule is left now, and now two molecules gap is formed after this reaction. Reaction number 5. This is reaction number 4, and the enzyme reaction number 4. Forget to write. Reaction number 4 enzyme is aldolase. Aldolase breakdown. Lysis reaction. Split into two half. One half from DAP, other half from GAP. And the reaction number 5, the DAP molecule now convert into GAP molecule, and now no DAP is there, and GAP molecule convert into 2. So afterwards, we written 2x, 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 2x. All things are 2x because that is converted into gap. The reaction catalyzed by, there is also isomerization reaction. This is ketose sugar. This is aldose sugar. Ketose to aldose conversion. That means another molecule of isomerase is needed, but the name is changed. The name is triose phosphate isomerase. Triose phosphate isomerase. A short name is TIM. TIM. T for triose, IM means isomerase. This is a first 5 reaction where glucose convert into 2 molecules of GAP, and these steps are, we know that it is a preparatory phase. That means prepare the glucose, modified the glucose by different 5 reaction, and now it will be benefited to give energy. Next up. Clear? Can you see during the preparatory phase already 2 molecules are ATPs involved? So preparatory phase for the preparation of glucose to give energy, 2 molecules of ATP already required. Clear? The next 5. Next 5. Payoff phase. After preparatory phase, we end 2 molecules of glyceraldehyde 3-phosphate and started with this 2 molecules of glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate, that means gap. In next reaction, that means reaction number 6, very critical reaction, where it convert into 1,3-bisphosphoglycerate, convert into 1,3-bisphosphoglycerate. Bisphosphoglycerate. Short name BPG. Bisphosphoglycerate. 1,3-BPG. Here also 2 molecule, 2x. All are in 2. Now look at the reaction here. Glyceraldehyde 3-phosphate. That means one phosphate is there in the carbon number 3. But next, after the reaction, 2 phosphate is there, 1 and 3 position. That's why these words came. So 2 phosphate means another phosphate is required, but this phosphate is not from the ATP, direct phosphate, PI, inorganic phosphate, PI I means inorganic phosphate. In the cell, we have phosphate pool inside the cell, and from the phosphate pool, Pi will become. The enzyme here, glyceraldehyde 3-phosphate dehydrogenase. Dehydrogenase. Short name GAP-DH. 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 Glyceraldehyde 3-phosphate dehydrogenase GAP-DH. Dehydrogenase enzyme not only added the phosphate group but changed the substrate by means of oxidation reduction. Dehydrogenase, all kind of dehydrogenase enzymes are involved in oxidation reduction reaction also. So here oxidation reduction reaction occurred, and the cofactor coenzyme needed for this is NAD plus. NAD plus, which convert NADH. That means NAD plus here reduced, and the substrate glyceraldehyde 3-phosphate here oxidized. So it is a critical reaction because here phosphate is added, and second, the product is also oxidized. And for the oxidation, NAD plus is required and NAD plus is reduced. The important step, reaction number 6. All are in 2x, 2pi, 2 NAD plus, 2 NADH. Because substrates are 2. Next reaction after TPG formation, 3-phosphoglycerate. 3-phosphoglycerate. Short name PG. 3PG. Look at the product. Substrate is 1, 3 and the product is 3. So one phosphate is eliminated. And the eliminated phosphate connect with the ADP to form ATP. So this is the reaction step where ATP is formed. How many molecules ATP will be formed? 2. All are 2. That means 2 ADP are involved. 2 phosphates are released and 2 ATP are formed. The reaction is catalyzed by phosphoglycerate kinase. Phosphoglycerate kinase. Short name PGK. PGK. From 3-phosphoglycerate, it converts 2-phosphoglycerate. 2PG. 2PG. Just change the carbon number of the phosphate from 3, it now becomes 2. This reaction is catalyzed by phosphoglycerate mutase. Phosphoglycerate mutase. Mutase means just change the phosphate number. Change the phosphate group numbering from 3 to 2. Short name PGM. Phosphoglycerate mutase. After that 2PG convert enolase enzyme and form phosphoenol pyruvate enolase enzyme form phosphoenol pyruvate. Another name PEP, 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 phosphoenol pyruvate. All are in two molecule, 2x. Reaction number 7, reaction number 8, reaction number 9, and the last reaction step where PEP is converted into pyruvate. PEP is converted into pyruvate. We reached the target. We started with glucose, and first we mentioned glucose is converting to pyruvate. After the 10 reaction step, it converts into the pyruvate, two molecule of pyruvate. Enzyme name, sorry, enzyme name pyruvate kinase, pyruvate kinase, PK. 10 reaction. Now see the substrate and product. In the substrate, there is a phosphoenol, that means phosphate group is there. But when the product is from pyruvate, no phosphate. So phosphate is getting eliminated from the substrate and enter into the ADP and form ATP. Same reaction done, which is in the reaction number 7. Phosphate is out from the phosphenol pyruvate and enter into ADP. So here also two molecules of ADP convert two molecules of ATP in reaction number 10. So two reaction step in the payoff phase, reaction number 7 and reaction number 10, where 2 ATP, ATP molecules are formed. So if you count the total ATP calculation, how many ATP will be formed? How many ATP? Credit and debit. Just look at that manner. Amount credit and debit. Here also ATP credit and debit. Credit occur in last half where 4 ATPs are generated, 2 ATP and 2 ATP. It is the credit amount, 4 ATP, and first half preparatory phase is a debit. That means it needed ATP molecule. So 2 ATP minus, so total ATP after the glycolysis is 2. So this is the first stage of any uh, respiration cycle, aerobic or anaerobic. Now pyruvate have the option. It can go aerobic cycle, aerobic respiration further, or anaerobic. It is the first stage, glycolysis. Without glycolysis, aerobic, anaerobic respiration cannot happen. Do not happen. Glycolysis clear all the reaction steps. Those are in online. Clear glycolysis reaction states.
Yes, sir. Clear.
Okay. Okay. So after glycolysis, pyruvate molecules form, and pyruvate molecules have two different fate. The next topic is fate of pyruvate. Fate of pyruvate. Fate means what happened after. What pyruvate wants to do. Fate. Pyruvate has two options. It can enter aerobic respiration, it can enter anaerobic respiration. Depending on the cell internal environment, pyruvate has two options. If pyruvate enter aerobic respiration, further pyruvate degraded by TCA cycle or Krebs cycle, followed by further electron is transferred by electron transport chain and oxidative phosphorylation oxidative phosphorylation occur to make huge quantity of ATP. Yes or no? And the amount of ATP is 38 molecule. Huge quantity of ATP, and that is 38 molecules of ATP will be produced if pyruvate enter aerobic cycle. Aerobic cycle needs much more lengthy steps. First enter into TCA, then TCA to ETC, that means electron transport chain. After electron transport chain, oxidative phosphorylation occur, and during oxidative phosphorylation, ATP will be produced. And the amount is huge. But pyruvate has another option depending on cell. It can enter anaerobic respiration. It can enter anaerobic respiration, that means it can enter fermentation. Fermentation, it is a quick process where acids or alcohols produced. Some additional step after glycolysis, one or two, usually one, sometimes it is two, one or two additional steps are needed and ferment the pyruvate into acids or alcohols. So fate of pyruvate can be of two depending on cell types and cell composition, cell internal environment, either oxygen presence or not. If it is present, aerobic organism, it enter into the this type of reaction process, TCA, ETC oxidative, and quantity of energy will be high. But the anaerobic respiration, just additional of one or two substrate, one or two reaction, where pyruvate convert into either acids or alcohols depending on cell. Normally, yeast prefer to produce alcohols, but bacteria prefer to produce acid. Types of fermentation, we will go further in next section. Clear, Tina? Any question? But I have one question and genuine question. Why some additional steps are needed after pyruvate fermentation in case of aerobic? Question. Why do cell fermentation occur? Why do cell fermented? Why do cell fermented pyruvate into acids or alcohol? Can you ever thought that why? If you
Because of
say it's a
microorganisms.
this another time because of
Because of microorganisms.
microorganism can be halted in the pyruvate also. Why extra steps are needed? They can be halted in pyruvate. Why need another steps, another 2 or 3 steps to make acids or alcohol? If you say it is the process of energy, we easily deny it because during pyruvate step we produce two molecules of ATP. During acid alcohols, no energies are gained. No extra energy is gained from the aerobic anaerobic fermentation, anaerobic respiration fermentation. But there is a logic. In aerobic, after two molecules, huge quantity of energy is gained. But what is the logic behind that? Why 2 or 3 steps are done after the pyruvate formation? Additional steps. Check the glycolysis reaction first. In glycolysis reaction, answer is remains. The answer is remain in glycolysis reaction. That's why I written glycolysis reaction in details. Check that. Quickly review the glycolysis reaction and try to answer. Check the reaction number 6 in glycolysis. Check reaction number 6 in glycolysis. I said earlier it is a critical step because in reaction number 6, the needed coenzyme is NAD plus. Needed coenzyme is NAD plus. And did you know NAD plus full name? Nicotinamide adenine dinucleotide. It's a full name. NAD plus actually the product of vitamin B3. Vitamin B3. NAD plus actually come from vitamin B3. So NAD plus important molecule in reaction number 6 and it came from vitamin B3. So in every glycolysis reaction, that means in every glucose reaction catalyzed state, NAD plus is required. If you do not regenerate the NAD plus, ultimately what happens? NAD plus will be eliminated or NAD plus amount will be, concentration will be less. NAD plus concentration less means no glycolysis. Further formation of glycolysis cannot happen because reaction number 6 need NAD plus and this NAD plus need B3, vitamin B3, vitamin B3. Vitamin B3 important molecule. Vitamin B3 is short, that means it needs for NAD plus. So vitamin B3 is less concentration, NAD plus will be less. So you have to be regenerate NAD plus. Generate NAD plus. Otherwise NAD plus will be shortfall. NAD plus will be shortfall means no glycolysis. No glycolysis means no energy production state. No energy production state means cells are die. Cells are die means tissue are die. Tissue mean organ. Ultimately organism totally collapse if glycolysis will be stopped because it is the major reaction scenario steps. So one of the important molecule during the glycolysis reaction is NAD plus and which come from vitamin B3. And vitamin B3 is limited inside the cell. It is a limited. It is not plentiful. It's a limited. So that's why you need to regenerate. And for this regeneration, bacteria will do these additional steps. So after pyruvate formation, it converted into acids or alcohol, and the main aim objective was to regenerate the NAD plus. Otherwise, glycolysis will be stopped and cells will be died later on. To maintain NAD plus concentration inside the cell, to glycolysis carry on, NAD plus have to be regenerated. That's why 1 or 2 additional steps are done by the bacteria where NAD plus will be replenished. In during fermentation, bacteria NADH during acid formation, alcohol formation, NADH will be converted to NAD plus. Check that. NAD plus will be replenished. So cell maintain the NAD plus now. Otherwise, glycolysis will be halted. Clear? Clear? Fermentation types. So why do cell need fermentation? For fermentation, because cell need fermentation due to NAD plus regeneration. Without NAD plus regeneration, no further glycolysis happen. That means no glucose is converted. Ultimately, energy reaction, energy production steps will be done, completed, and that means cells will be died later on. So this will be critical NAD plus regeneration. And in aerobic cycle also, during TCA cycle, etc., NAD plus is regenerated during TCA cycle. Check the cycle in the book. The TCA cycle, NADH to NAD plus is generated. From NAD plus and NADH is generated, and during ATC, NADH, NAD plus is converted. NAD plus is not required. Otherwise, glycolysis stops soon. It's come from the B3, vitamin B3, and vitamin B3 is not, it's a limited concentration inside the cell, always in limited. That's why NAD plus should be regenerated. So fermentation, acids and alcohols are produced bacteria or other organism due to NAD plus regeneration, and it benefited our in humankind. That means we can use acids and alcohols into variety of to make variety of product, variety of fermentation product. That reaction state will benefit our to improve the product. That means fermentation process. So fate of pyruvate done, and next task, types of fermentation. Types of fermentation. Two types based on the product formation. Number one, alcoholic fermentation. And number two, lactic acid fermentation. These are the two major types. In alcoholic fermentation, it is done by some bacteria, mainly yeast. Alcoholic fermentation done by bacteria and yeast, where yeast converted pyruvate structure, 3 carbon structure, C double O minus, C double bond O, CH3. It is the structure of pyruvate. Pyruvate converted first acetaldehyde with the enzyme pyruvate decarboxylase. Pyruvate decarboxylase. And pyruvate decarboxylase need TPP, thiamine pyrophosphate or coenzyme. Pyruvate convert into acetaldehyde, CH3CHO, acetaldehyde. Now came the second step, crucial step, where NAD plus will be regenerated from acetaldehyde to alcohol, CH3, CH2OH. ethanol. This reaction is catalyzed by alcohol dehydrogenase, and it is the state where NAD plus will be regenerated by the help of alcohol dehydrogenase. One molecule of NADH convert into NAD plus, which is the primary criteria of fermentation. NAD plus regeneration. Check that. So during alcoholic fermentation, NAD plus will be generated and ethanol will be byproduct. We can use this byproduct in our work to manufacture wine, beer, etc. Here, alcoholic fermentation, reaction step, two-step reaction, two additional reaction step to convert NAD plus. Primary motto is this, bacteria. This is the byproduct, ethanol. Primary motto to convert NAD plus. Lactic acid fermentation. Lactic acid fermentation done by bacteria majorly, and the group of bacteria are called lab. Full form of lab, lactic acid bacteria. Lactic acid bacteria, a group of bacteria that actually produce lactic acid during fermentation. Lactobacillus, one of them, lactic acid bacteria example. Lactobacillus, where we can find this lactobacillus, where we can find lactobacillus, curd, correct. Curd contain high quantity of lactobacillus. So lactobacillus is a one example of lab. Bacillus, etc. Lactic acid fermentation. In lactic acid fermentation, alcohol is not produced, it is produced acid, and the acid is lactic acid. So pyruvate structure, C double O minus, C double bond OCH3. It's a one-step reaction. Just one-step reaction where pyruvate convert into lactate. Lactate structure C double O minus CHOH CH3. In single step reaction, lactate will form. It is the not major product. The primary object of the of this reaction for bacteria, bacterial scenario. Our scenario it is lactate, but bacteria scenario to regenerate the NAD plus. In this reaction, NAD plus will be regenerate. NADH convert to NAD plus, and the enzyme that converts is called lactate dehydrogenase. Lactate dehydrogenase. Short name of lactate dehydrogenase LDH. We can further classify the lactic acid fermentation into two types. This can be further classified into two types: homolactic acid fermentation and heterolactic acid fermentation. Homolactic acid fermentation, and heterolactic acid fermentation. Check the word homolactic acid. Homo means, check the word hetero. Hetero means good. Homo means same. Hetero means different. That means in case of homolactic acid fermentation, we know that here only lactic acids are formed. Only lactic acids formed. So these bacteria only convert pyruvate into lactic acid. And we mention over there lactic acid bacteria, bacillus. These are two bacteria only convert lactic acid, and the reaction is this, from pyruvate to lactic. So this reaction actually homolactic acid fermentation reaction. But in case of hetero, we retain here different. That means not only the lactic acid, but also alcohols are also generated. In case of heterolactic acid fermentation, lactic acid produced as well as ethanol, which is the actual product of alcoholic fermentation. Ethanol also generated in heterolactic acid fermentation. So two molecules are generated and two different molecules. That's why the name is heterolactic acid. But in that case, only lactic acids are generated. That's why same homo. Now we find out why two different molecules are generated in heterolactic acid fermentation. The reaction cycle. Next topic, heterolactic acid fermentation. Heterolactic acid fermentation done by some special bacteria like leuconostoc, not the lab. Lactic acid bacteria only form homolactic, but leuconostoc can do heterolactic acid fermentation. And these bacteria actually helpful to improve the taste, the texture, the flavor of the product. We also discussed in latter unit 2 to 5 where the fermented foods are there. Here also we can discuss this importance of heterolactic acid fermentation, importance of leuconostoc. Leuconostoc actually improve the taste, texture, and flavor of the fermented food. Fermentation basically done by lactic acid fermented bacteria lab, but it's only single test. That means pH low and it's taste like acidic. But further improve the taste, further improve the texture, we need different product formation. and that will done by heterolactic acid fermentation. It has very important role during the fermented food. That is the topic. Fermented food production, texture, flavor, and taste. So how do that? The reaction, two different products are generated in heterolactic. Check the cycle. First, glucose converted into glucose 6 phosphate. They are done by some different way. P circle means phosphate. From glucose 6 phosphate, in glycolysis, it enters into the glucose 6 phosphate, but here it changes into shuttle into 6 phosphogluconate. 6 phosphogluconate. Just bypassing the step of glycolysis and enter into the another step where they can produce two different types of products. Glucose 6 phosphate, the reaction is same, that is in glycolysis, but after glucose 6 phosphate production, they change into the 6 phosphogluconate. They do not produce fructose 6 phosphate. If fructose 6 phosphate is produced, that will enter into the glycolysis. But change into the 6 phosphogluconate. This 6 phosphogluconate further convert into ribulose 5 phosphate. Ribulose 5 phosphate. And from ribulose 5 phosphate, xylulose 5 phosphates are produced. Xylulose 5-phosphates are produced, and xylulose 5-phosphate break down, split, split into GAP, glyceraldehyde 3-phosphate, and acetyl phosphate. GAP thereon convert the next half of the glycolysis, that means payoff phase reaction cycle is same. That means gap convert into 1,3-BPG, 1,3-BPG to 3-PPG, 3-PG, 3 to 2-PG, phosphenol pyruvate, then pyruvate. So gap converted like the same reaction cycle that occur in the payoff phase of glycolysis and enter into pyruvate. From this pyruvate, they convert lactate. Here NAD is convert into NAD plus, regeneration done. And from the acetyl phosphate, they convert acetaldehyde, and from the acetaldehyde, alcohol dehydrogenase. What this alcohol dehydrogenase do? It convert acetaldehyde into ethanol. That is the alcoholic fermentation step. Acetaldehyde convert into ethanol. Here also NAD plus can be regenerated. So check the pathway. In heterolactic acid fermentation, it not usually go in glycolysis, but change the shuttle into different pathway. The pathway is known as phosphoketolase pathway. Phosphoketolase pathway. Change from glycolysis, it enter into phosphoketolase pathway, and from there on, they produce two products. The divergent points, glucose 6 phosphate. Divergent point, glucose 6 phosphate. Glucose 6 phosphate usually go into fructose 6 phosphate in glycolysis, but they diverge into 6 phosphogluconate, and thereon they produce ribulose, xylulose 5 phosphate, and produce lactate is the byproduct. NAD plus is converted, the primary thing is done. Here also ethanol is produced from the acetyl phosphate. Here also NAD plus can be regenerated. So two products are generated in one reaction state. That's why it is heterolactic fermentation. Important bacterial group that turn leuconostoid. Any question from today's session? This is end here. The first topic ends. That means basics of fermentation. We discuss today. We discuss fermentation meaning. Fermentation. Why fermentation? Do by the bacteria. Why fermentation? And next আমরা ডিসকাস করলাম fermentation types. Any question? না হলে গ্লাইকোলাইসিস হবে না তো। So important NAD plus কে regeneration করা, and we are lucky for that. Otherwise our fermented products will be incomplete, cannot be generated. NAD plus is not required, then check pyruvate. After pyruvate the reaction will be stalled. Do not need any additional step. Why bacteria will done that? It is done for only NAD plus regeneration, and we are lucky. যেটার জন্য আমরা অ্যাসিড ইথানল তৈরি করাতে পারছি, ফুডের টেস্ট টেক্সচার চেঞ্জ করাতে পারছি। And another statement is for product is during alcoholic fermentation. I think we forget to read, right? Alcoholic fermentation product is ethanol, but there carbon dioxide also produced, CO2 is also produced. That Janvi told before this class during the fermentation, she said about the dough is puffed up by means of carbon dioxide production. By means of carbon dioxide production during the yeast, when alcoholic fermentation done, carbon dioxide also generated, and this is also helpful in further improve the fermented products. Next day topic from this unit: microbes involved in fermented food production. Next day topic. That means next Saturday. Microbes involved. We see in this session that also microbes, that means bacteria, lactic acid bacteria, and in alcoholic fermentation some bacteria, yeast also involved, and there are other bacteria also involved, other microbes are also involved, and that will be discussed in next class. Microbes involved in fermented food production. Next day.